This is the Chapter 9 Proteins Part 1 commentary. As we continue on, we're going to go ahead and sketch crude polypeptides. So first thing I'm going to go ahead and do is go back to my Chapter 9 Proteins Part 1 slides. What you absolutely need to do first is memorize. Make sure you memorize these 4 amino acids. So I could ask you to sketch a dry, for example, aspartic acid. In which case you have to go ahead and draw it. Next, I could sit there and change it up and ask you to sketch an aqueous aspartic acid. Keep in mind, aqueous means it's dissolved in water. So, for example, you're going to have to go ahead and turn this into a positive ammonium, and then you'll have to turn this into a negative carboxylate. So let me clean up that last one over there since it's harder to read. There we go. So in this case, it's a negative carboxylate. So now we have our zwitterion. The only other thing I need to change is the very bottom, because this is a acidic amino acid. It's going to give off its hydrogen, forming a negatively charged carboxylate anion. So even though these are balanced out, the very bottom is going to have a negative charge. Now usually we don't see this floating around. There is other positive, such as partial positive from a nearby solvent molecule, from a hydrogen to balance it out, or there are floating sodiums or potassiums that are actually dissolved in your cytoplasm, for example, of a cell, and they'll help balance it out. You don't have to sit there and put that. Bottom line is you'd have to sketch everything you'd see here if I asked for the aqueous. Now, most likely what's going to happen is I'm going to ask you to combine these into polymers. Polymer is a general term. After the condensation reaction, they'll either form a polypeptide or a protein. Remember, a polypeptide is a polymer with less than 50 amino acids that are connected with peptide linkages. Protein is more, but I'll just use the generic term polymer. I could give you a problem like this and say something like, say something like this and say, sketch a 3 amino acid, forgot that famous word, aqueous residue polymer. Now, if you go ahead and you take a look back at the other slide, under it, for example, under aspartic acid, you'll find other symbols. You'll find a ASP. You'll also find a D. So these are the 3-letter notations which represent aspartic acid. This is the one-letter notation, which represents aspartic acid. Keep in mind, biochemists, we're dealing with these amino acids all the time. So we get lazy after a while, and we need shortcuts for naming these. You'll be responsible for naming the shortcuts. So I could give you a problem like this and say, okay, ASP, LEU, LYS. The first one is for aspartic acid, the second one is for leucine, the third one is for lysine. Or I could give it to you in this format, D, L, and K. Again, D is for aspartic acid, L is for leucine, K is for lysine. So memorize everything on that slide, both the sketches, the names, as well as the 3-letter and 1-letter symbols. So let me go ahead and erase some of this just to create extra space. So I'll give you something like this. ASP, and I'll stick with the one I just did right now, LEU and LYS. There's many ways you can do these. You can first, having memorized them, simply write out the non-aqueous individual amino acids and then link them together. Or you can just simply write them out in their final form, whichever is easier for you. Some of you can mentally picture these. Others need to write them down on paper, whatever it takes to get to the final answer. So let me just go ahead and write the non-aqueous for right now, and then I'll go ahead and write the final aqueous polymer down below. So for aspartic acid, we have H2N, carbon with its hydrogen, CO2, CH2, and then COO, OH. Here we have leucine. Leucine looks like this. H2N, CH, and then in this case for leucine, because I know how it's going to look, I like to get a little bit lazy on this, and you can too, provided you use proper line angle structure. I can sit there and create something like this. Alright, and then you have the rest of it. CH2, whoops, forgot my other one. Getting a little messy there. And here we go. And then finally for lysine, we have this over here, H2N, CH, COO. Again, for lysine, I'll make it a little lazy. It's got 4 carbons on the alpha carbon. 1, 2, 3, oops, 1, 2, 1, 2, 3, 4. And then we have our NH3. Whoops, I screwed up. Why did I screw up? Because technically I'm still drawing the non-aqueous form as a starting point. So here's my starting point. And we'll do NH2. I put the hydrogens on the left-hand side. You can put them on the right-hand side if you want. Now, having memorized that, what you can then do is convert it into its aqueous polymer. The first thing we're going to go ahead and do is change our left-hand side. The left-hand side is going to be the N terminus. So this is the N terminus. and it's going to be an NH3+. Technically, this hydrogen comes off, so do these hydrogens over here. But since we're drawing a polymer, we'll just go ahead and actually, let me make a bigger circle right here. It's actually, so the OH from that carboxyl group and a single hydrogen is going to come off, forming a water. It'll then look like this. I'll scratch that out. I'll go ahead and I'll move the hydrogen on the bottom. Let me go ahead and change pen colors. So I'm just going to put the hydrogen down here, and then here we have a really long line. Oh, there's an old lone pair. Scratch that out. Let's go over to the other side. Again, the OH from the carboxyl group, one of the hydrogens, is going to go ahead and form a water. So let's scratch this out. Scratch out the hydrogen. We're going to go ahead and put the hydrogen on the bottom, just like last time. So let's put it down here. And then I'll just draw a line going straight across. I have a thicker marker, but that's okay. Now, the C-terminus, that's the end amino acid residue, which is now going to form a negatively charged carboxyl ion. So we erase this right here. We then have our leftover lone pair. Here we have a excess negatively charged. So here's our negatively charged carboxylate ion, and this last portion is called the C-terminus. Notice we have the N-terminus, the C-terminus here, and then we have our very last change which we need to make, and you need to go through each of these side chains. Are the side chains acidic or basic? If they're acidic or basic, you need to make the necessary changes. For basic lysine, it gains a hydrogen from the nearby solvent, forming a positive ion. So this right here is what you'll be expected to draw, and you can go straight to the final product if you want. You don't have to go through the steps that I did, but this will be the final product that you need to draw if I give you the starting 3 amino acids. So this is the proper answer for sketching a 3 amino acid aqueous residue polymer. Make sure you memorize everything that's on this slide. The sketches, the name, the 3-letter symbols, and the 1-letter symbols. Again, these are only 4 out of 20, so not too bad. If you were to take a traditional biochemistry class, you have to know them all. So that said, you're then going to have to sketch future ones showing intermolecular forces like you see here. In the dry lab / in-class discussion, you're going to be given a paper like this. And you're going to be asked, given these starting amino acids, you're going to be asked to create the aqueous polymer amino acid residues. Basically, you'll be asked to create a polypeptide. And you're going to have to emphasize key side chain intermolecular forces. So that way you can really appreciate the intermolecular force stickiness as these polypeptides loop around themselves to form their 3-dimensional structure. So let's go ahead and take this one right here. I'm going to go ahead and copy this. I'm going to go ahead and create a temporary new slide. And let's go ahead and create this one. This one takes a lot of time. This is really hard. My advice is to first master each of these, and then go ahead and first master these, and then go ahead and start drawing them the way I do here. The first thing you need to go ahead and do is create your backbone. Notice we have 1, 2, 3, 4, 5, 6, 7 amino acids, which are going to form residues in our polymer. So each of them have the backbone NCC. So NCC, NCC, that's number 2. NCC, that's number 3. Now, what's going to happen is you're going to create a what we call a hairpin polypeptide. And it looks just like a hairpin. It's going to double back on itself. So NCC, and this is where you're going to have to start turning the paper. And just so I don't lose count, that's 1, that's 2, that's 3, that's 4. And then as I stand up to draw the rest of them, because I'm terrible at drawing backwards, N, C, C, that's 5, N, C, C, that's 6, and N, C, C, that's 7. So 5, 6, 7. I then go ahead and number up top. Now, from here, I know the ASP is aspartic acid, SER serine, leucine, lysine, leucine, serine, lysine, and so forth. We know that the N's are always going to be the N-terminus, and the very last amino acid residue is going to be the C-terminus. And just because it's cysteine, there is no connection. It could be a lysine at the very end. It would still have a C-terminus. So I'm going to go ahead, use a different color. I'll use blue. So first thing I will do is go ahead and put a positive right here, NH3 positive. That's my N-terminus. And I'm going to go ahead and fill in the rest. This has a negative charge. That's my C-terminus. Now, the next thing we're going to go ahead and do is I'll start putting in all the rest of the backbone filler, as I call it. And you can do it in different ways, but this is how I do it. So I'll probably just go ahead and use a, which color should I use? Oh, let's use a green. So all the alpha carbons, those are the center carbons, are always going to have a hydrogen on them. And then the carbon next to the alpha carbon is always going to have a double bond. And then finally, each of these hydrogens, each of these nitrogens will have a hydrogen on them. Now, regardless of which amino acid residues you have, this backbone shape will always be the way it looks here. It'll always be the same. The amino acids represents the side chains that are present. And therefore it represents what dominant intermolecular force side chain interactions are occurring. So you should be able to write this backbone really easy Make sure you become very comfortable with it, going over it over and over and over. In this case, I have 7 amino acids, so it would look like this. You need 7 amino acid residues. Now you start filling in each of the side chains. The aspartic acid is going to go ahead and have a carboxylic acid that's present. Note, keep in mind, because it's aqueous, it loses the hydrogen. We go over here to the serine. Serine has, oops, screwed up on this one. I forgot to put in my carbon. Let's go back to this one. So here we have CH2OH. Next one, carbon 3, we have a leucine. Again, I'm just going to go ahead and get lazy and draw the line angle structure. Next one is going to be a lysine. Now, usually you have the leucines on the inside. The nonpolars like to stick next to each other. They stay away from water as much as possible. The lysines, depending on whether there's a long-range intermolecular force or not, you can have them on the inside, or you can have them on the inside outside. Usually you have them on the inside if you're representing these salt bridges. It doesn't matter to me which side of the alpha carbon you have them. For this one, just to keep things simple, I'm just going to go ahead and put it on the inside for right now. Keep in mind it's a positive charge. Notice, realistically, it would probably be on the outside because it'd want to be as far apart from that leucine as possible, because leucine is nonpolar and the lysine is very polar since it's a ionic positive. Either side, though, I will give you full credit. Let's go over to this one over here, NCC. This is carbon 5. That's going to be a leucine. You know what? That one's really bothering me now. A, I need the space, and B, it really should be on the outside. So I'm just going to draw it on the outside. Notice it's a positive charge. So I'll cross that one out. All right, carbon 5, same thing, leucine. So here's that hydrocarbon tail. Let's go to number 6. That's going to be serine. Serine. So in this case, here's our alpha carbon, CH2O. H. And then finally, the last one is cysteine. Now, the next thing I'll do is I'll ask you to name some characteristics. So, I've designed the characteristics so that if you draw the hairpin polypeptide in this format, you should be able to show these characteristics pretty easy. I will usually ask you to indicate what is the dominant intermolecular force. So going to a purple pen, in this case, if I ask for show me a hydrogen bonding side chain intermolecular force, you have to find 2 amino acid side chains which can contribute to hydrogen bonding. In this case, if I have 2 serine, because there's a lone pair and a hydrogen attached to a nitrogen, oxygen, or fluorine, in this case oxygen, then hydrogen bonding could occur. You don't have to get crazy on how you associate them. Just simply draw a circle or a dash and indicate hydrogen bonding. Are there other hydrogen bonding that is present? Yeah, if you wanted to. If this carboxylate was far enough over, you probably could have showed hydrogen bonding on this one. So I'll give you some leeway on this, provided the side chains can participate in a intermolecular force that I indicate that I want you to emphasize, in this case, hydrogen bonding. Notice, serine could not participate in hydrogen bonding Why? Because the hydrogen and lone pair are not on a nitrogen oxygen or fluorine. It's on a sulfur. So the most that this would do is dipole-dipole, and I'm not indicating that in this example. I could ask other things like go ahead and classify each of these as polar, nonpolar, basic, or acidic, and that's what you have to have already memorized. So this first one, carbon 1, is going to be aspartic acid is going to be acidic. Oops, acidic. This next one over here, carbon 2, is going to be polar. The next one over here is going to be nonpolar. The next one over here is going to be basic, nonpolar, polar, and the last one is going to be polar. Cysteine, please keep in mind, although it's not part of the top 4, you must absolutely know cysteine. So this is the additional one. Again, cysteine looks like this, drawing its aqueous amino acid form, positive CH, COO. So it's got a CH2SH. So that's a cysteine. That's a cysteine. So even though the slides say no 4, you have to know all 5 amino acids and apply them. So make sure you know that 5th one, cysteine, as well as the other 4. So in this case, in the lab, you'll be asked to do many of these types, and then you can pretty much guess how I might ask you on an exam the theory behind amino acids and intermolecular forces. I'll ask you to do exactly what I've just demonstrated here, so make sure you know this. So that concludes the part 1 set of slides.